Okay, welcome everyone to this next episode of Trainee Pearls, a podcast from the EBMT Trainee Committee. I am Lars Gerde, a hematology and BMT trainee from Rigshospitalet in Copenhagen, Denmark. And I'm very happy to guest host today's episode, where I am very happy to be joined by Dr. Oscar Brück from the Department of Hematology and the Data Administration Department at the Helsinki University Hospital in Helsinki, Finland. So thank you very much, Oscar, for accepting our invitation.
Thank you, Lars. Thank you for having me here.
Yeah, it's a pleasure. How are you today?
I'm well, thanks. Doing some research and some applications by the side as well. So yeah.
Excellent, excellent.
Excited to discuss the publication today.
Yeah, that's why we brought you in. So recently you published in Blood Cancer Discovery this paper entitled Machine Learning of Bone Marrow Histopathology Identifies Genetic and Clinical Determinants in Patients with MDS. And congratulations with the publication.
Well, thank you. Thank you.
So to start things off, could you talk a little bit about like the diagnosis and classification in MDS, and which research questions your study wanted to address in relation to that?
Okay, well, that's a quite large question to start with, but I'll try to answer that. So, MDS is a quite heterogeneous disease where hematological patients usually are admitted to the clinic with some kind of a disorders in their complete blood count, and symptoms that they can have can be related to anemia. So, for example, fatigue, shortness of breath. They can have some infections, or they can just feel very ill. And the diagnosis is based on WHO guidelines, and they include identifying cellular dysplasia from bone marrow aspirate or biopsy samples, and as part of the diagnosis, and also to risk stratify the patients, the karyotype, the blast proportion, and then also the complete blood count are assessed, and then as to get an idea of the prognosis, the IPSSR, so the revised international prognostic scoring system, is calculated for patients, and that is also part to guide the treatment as well.
Right.
And so now, if I got the other part of the question, can you repeat?
Oh, thank you very much for laying out that, so we all know how the current status is in diagnosing and classifying MDS. So how did your study, which questions did you want to address in relation to that?
Okay, so yeah, so we started off we're very excited by recent papers at the time that studied whether the presence of individual mutations and cytogenic alterations could be detected from very routine biopsies. And those papers were done in lung cancer by Nicolas Coudray, and then by Jacob Kater in colorectal cancer, and there were some others as well. And the findings were quite striking, that just by looking at the H&E stainings, you could really detect those individual mutations in some cases quite accurately, actually. And we just quite asked that could we do the same in MDS? And so we collected bone marrow biopsies of MDS and MDS MPN, so MDS patients with myeloproliferative neoplasm syndrome, and also healthy controls as a reference. And we sequenced diagnostic samples that were stored from these patients as well with a clinical sequencing panel, and we collected their karyotype, and the tools that we use to mine those images are basically two sort of tools. We use convolutional neural networks to really mine the images, so to extract the morphological features of those images. And then we used multivariate penalized regression models to build those individual models to predict the individual mutations and cytogenetic changes. And well, you can read on the paper that we got actually very good results to detect individual mutations and individual alterations. And we also tested then, could we predict things like the age of the patient and the IPSSR score of the patient and prognosis and so on. And basically we could predict all of those really well, and that's really fascinating to see that the routine stainings have a lot more information that is currently being used in the clinics.
That's very interesting. So you said you used convolutional neural networks. That is like a way of saying that that's the part of one part of the machine learning or artificial intelligence, or what you would call that. So was that something you developed yourself?
No. So for the convolutional neural networks, we used to go quite deep already, but it was basically pre-trained models. So models that have been trained in large data sets of millions, 20 million images that were, and basically the model parameters were so that were used to detect, for example, dogs and cats and houses and humans from those images. And we could use the same parameters here to analyze the H&E staining, so to identify the different morphological and texture features from the images.
Oh,
so we just basically used off-the-shelf model without any, we didn't really change the parameters there, to really just get out of the images what you can get with that anybody else can replicate without basically large computing powers.
It's fascinating that you were able to build upon something that was built from a totally different background, but applying this on very complex histopathological samples. So just a side note, were these things free for you to use? Was it part of like an open software, or did you have to pay for this?
It's free to use. So we used open source libraries. So in this work, we used a library called Keras that is based on the TensorFlow framework, and that is anybody can use that, download that, and download the parameters as well. And that's really a great thing about the computer vision community, that these parameters that are very challenging to compute are also published for anybody to use to their own research work.
Mm-hmm. And I also saw that all the codes and all the different technological details have been also laid out freely on your webpage. So if anybody's interested in details on that, so I also commend you for also bringing that open science in your own work. So again, perhaps for people that are not that familiar with the machine learning and artificial intelligence as you are, just before we go into the specific results, can you say a couple of words about how machine learning and artificial intelligence differs from traditional statistics and some more of the definitions there used?
Okay, so usually machine learning is interpreted as part of the artificial intelligence, besides other non-machine learning based statistical models, just regression models. Personally, I have some maybe challenges to really understand what artificial intelligence should be really interpreted, but machine learning is in a way much easier to understand, I think. So it's the study of computer algorithms that can improve by using data. So you build models based on a subset of data called training data, and you kind of try to develop or train some sort of parameters. And when those parameters learn the representation of the data, or let's say like the phenotype, let's say if is it like survival, or if it's like the occurrence of a mutation or whatever, you can then test how well those parameters then work in a separate data set, or test data set, or validation data set. And you can make it more robust by really testing in different data set from different data sources. For example, in the clinical context, it will be from different hospitals, for example. And and there are many different frameworks to train machine learning algorithms. The last decade has been really the era of deep learning more and more, especially in text mining and image analysis, and the performance have been often even superhuman, and that's really an exciting thing that we can build tools from humans to the human to make the clinical work, for example, less tedious and maybe easier, and more maybe someday even more reliable as well.
Yeah, I agree. I think we also start to see many applications now within hematology and bone marrow transplantation. So I think that we as clinicians, we might not need to know the deep ends of the programming and the techniques, but we need to have an idea of what these things are and what they represent and how they work. So going back to your study, when you applied this algorithm normally used to divide cats and dogs on these MDS and MDS-MPN histopathological H&E stained samples, you first use some sort of unsupervised learning methods where the computer basically just sees, okay, which patterns are there in these samples, and what did you find there?
So that's a very good question. So we used unsupervised learning first to analyze very small tiles or patches of those stainings. And the idea there was to understand, can we just see what is the bone marrow biopsies made of? What is it composed of? And we used UMAP projection just to divide the data into two-dimensional format. We used phenograph clustering, so a clustering method to divide the individual small tiles, so the small textures, and also a slingshot trajectory analysis to understand in a way, is there any kind of a pseudotime or some kind of a trajectory between those different textures? What is the kind of a connection between different texture subtypes? And to really, if I can more maybe explain, so we clustered and mapped here the image feature data. So one image is kind of composed of, in this example, we use 2048 variables or visual features, and that was the data we used to cluster and to understand what is those individual texture or small tiles composed of.
Interesting. So were you able to differentiate between patients with MDS, MDS-MPN, and healthy controls?
Yeah. So just by looking at the individual tiles, we're not able yet to differentiate. But then when we combined all the tiles from one sample together, then we were able to differentiate, especially the healthy controls. But among the MDS and MDS-MPN patients, there were some significant overlap, but we could also really see that some clusters that were more MDS-MPN specific, especially like the hypercellular samples, and so the unsupervised learning could really differentiate partly the overlap between MDS and MDS-MPN patients.
I see. So would you think, could this be a way of assisting the hematopathologist in their review, or can you replace the hematopathologist in differentiating between these things, or where are we with the technology?
Oh, I would say that not replacing, but I really, really hope that someday that will be an easy to use tool for the hematopathologist to analyze images. So to gather in an automatic, standardized way data from those images um, that would otherwise take more time or would be prone to subjectivity or would have other caveats. So I'm sure that that day will come, and I'm really eager to see how that can someday help the hematopathologist in his or her work. Yeah.
And one thing in machine learning, you say here, so you could cluster these things. You can see how the more advanced algorithms could find the patterns in the data that the human eye cannot do in these high dimensions. But another thing you did in your paper was that you tried to see if any of these morphological features could predict mutations, karyotype, prognosis. Can you say a little about like in the human eye, or do we have some histopathology and karyotype mutation correlations we already know? And did you find anything new, or could you predict anything else in your study?
Yeah, we know already that, for example, that MDS patients with that belong to this classical deletion 5q syndrome. So patients that have in the karyotype a missing chromosome 5q arm have hyperlobulated megakaryocytes. And that was something actually we could also find in by using our tools. And there are certainly many other associations that we have not really been able to find yet, and could be very exciting to find someday. And that was something that we really tried to answer in this study as well. So first, as said, we tried to we just wanted to see if we could predict the occurrence of those individual mutations and cytogenetic changes. And for example, for genes such as TET2 and SXXL1, we could find really a strong prediction accuracy. And then we tested that, is that prediction accuracy, does it correlate with the variant allele frequency of those mutations? So the real burden, so to see how many cells really have that mutation in the sample. And for example, for ASXL1, the correlation was really strong. And that was kind of an exciting validation that our platform works, or a model works in finding the relevant features in the images. And the last step was then kind of we want to see, can we really explain what is the reason why we could find those mutations? So if you still can remind that we have the convolutional neural network based method to mine the images, but then we did another approach where we detected cells and the third, and then also extracted features such as nuclear size and how dark the nuclei were. And the third image analysis method, where we just analyzed what kind of a texture features there were in the images. And by texture feature, I mean the amount of red blood cells, the amount of nucleated cells, amount of stroma, and bone trabeculae, and the amount of lipid droplets. So the kind of a synthesis was that we have the models to predict the mutations, that is based on the convolutional neural network data. Then we have the cell segmentation, the cell detection data, and then we have the pixel analysis that is trying to quantitate the amount of different textures. So we can then explain that, hey, we find that mutation quite reliably, and we can see that the prediction accuracy correlates with the size of, for example, of the nucleated cells, or with the amount of stroma in the sample. And in that way, we can in a way explain that most likely the machine is seeing those features in the images as the correlation is pointing to that direction, that there is more stroma and bigger nuclei, and that is correlating with the occurrence of a certain mutation.
So this is, correct me if I'm wrong, but this is a way to get more interpretability out of the algorithms into the user, for the clinician or the pathologist to understand more what's coming out of the so-called black box.
Yeah, exactly. Yeah, so that the kind of the explainable AI is really a hot topic at the moment, and I think we offer one possibility how to explain these models, what they're seeing from the images.
So how has your hematopathologist colleagues received your work? What do they think? What are the perspectives?
Yeah, so one of the co-authors is a hematopathologist, and he was really excited about the results. Of course, there is, even though we try to really explain what the machines are seeing, it's not like really explicit in that manner that we can say really at the exact things what the machine is seeing. But I think it's a step forward, and he was really excited to have that. Be part of that step, and I think we need a few steps more to to make the hematopathologist really happy.
So Oscar, you're also an MD and a clinician who have, but you always also obviously have a lot of insight into this computing and the technologies behind machine learning. So from where we are now, I understand that you would like to work on further on this in your own research. What is needed to be done in order to bring this to the clinic, and which implementations in the clinic do you foresee for this technology?
Excellent question. So there are some limitations to that study, and to list some of those, one is that the image digitization is made with 20 time magnification, and the stainings are H&E stainings, which are good to understand what is the bone marrow made of, but not optimal to see subcellular or even subnuclear features of the cells. And the data set, I think it's a nice data set, but it could also be larger. So and that's really difficult. That would mean a multi-center collaboration to get a really large data set. And but those three kind of challenges we are now currently trying to solve. And to do that, we are now digitizing every sample we have. The Helsinki University Hospital has collected and has stored. There are in total 60,000 samples in our storage, and we're digitizing all of them. And we are then developing new tools to analyze those images, and we are combining the image data then with clinical data that we get from the hospital database called the Hospital Data Lake. And that's something I've been working now with the data lake. I've been working a couple of years, and I think we are very far in getting kind of in getting all the data. So I mean, by all the data, I mean all the lab data, all the treatment data, all the diagnostic, whatever you can think of data together with the image data. And I think that will be we'll understand much better now what is the really significance of small morphological abnormalities, or the composition of the exact composition of the bone marrow cellular profile, so we can really do things that kind of have eluded now for 100 years the human eye, because the images have always, or the slides have always been there, but analyzing them in that scale has been impossible without like tools that we have now today.
That's impressive work. So you think, I think you said also that this techniques on especially images has been a very interesting field for data analysis using machine learning, given the vast amount of information that can be found in such images and within the medical field. And many of these have been relied on classifying or diagnosing conditions. Do you think that this is the best way to the best path to implement machine learning, or do you also see a role for it in predicting disease course and survival, for example?
Yeah, so both of them are kind of a part of our scope. That's really another important field. So to really identify biomarkers of treatment and biomarkers of prognosis. If we think about like the current clinical scores that we have in different leukemias, I think the image or the morphological data is actually quite limitedly used. And one of the reasons is that the examination is slow and it's subjective, and we have we around the world we're using this quite standard May, Grunwald, Giemsa staining or Romanovsky staining, but still the samples differ a bit depending when they're coming from, and I guess that we can really ameliorate the scores we have today by implementing high quality image data into, and it can be like numerical data from the images, but it can also be like the number of, for example, the numbers for different cell types that we can, like anybody can count, but it takes ages to count. So now we can do it in just a few minutes or even a few seconds, for example, and that could be then part of the standard diagnostics and monitoring of patients someday.
Right. So, and in speaking into that also, I think a lot of people have discussed the ethical aspects of employing machine learning and artificial intelligence. Can you comment a bit about how you have thought about that, or do you have any initiatives for addressing this at your in your research?
Yeah, that's an excellent question as well. So. So ethical and maybe also methodological issues related. So I would say the first one would be like selection bias of the data sets. Often in machine learning, the data sets are collected from patients that have been part of a trial or otherwise have had perhaps have the possibility of getting a treatment that is not accessible for everybody. So that imposes, even though we do not want to impose that selection bias, that I'm sure imposes a bias in having a distinct and not maybe representative patient cohort in terms of ethnicities and also like genetics can be. One issue is that in machine learning in general, I think people are very eager to develop models and show how well they work in their data set and perhaps in another data set. But I think that really to make the clinical impact is to continue developing that model in prospective trials. So that's the only way to get that model into the clinics or the clinical practice. So I'd say it's a general issue that one should be more interested in if you really truly believe in that. So we should really continue working, getting that part of the clinical routine work. And as an example, I mean, personally, I think, for example, RNA sequencing it's a huge resource for research, but still, I found a lot of different machine learning models that have been using RNA sequencing data, but there are not that many of them that, or I cannot think of any of them that is kind of currently used in the clinics, at least here. So I think there is a huge gap between the research kind of machine learning and then the clinical machine learning.
I agree, and I think an important part of acknowledging that issue and pushing that transition is to also have an interaction between those that know the technology and the programming and clinicians, and you yourself are a good representation of that, being both an MD and now also a PhD, having done work in this very complex field. So just at the end here, Oscar, how did you get into machine learning, and how would you recommend, how much should clinicians know about it?
It's a very huge field first. So I really encourage anybody that has interest to dive into the world of bioinformatics. For me, perhaps there were a few factors that led me into it. Around, let's say 5 years ago, I didn't do anything in bioinformatics, but I felt that the ones that knew how to analyze large amount of data were just too busy. So I was really like forced to get into this. So if I really wanted to use machine learning in my research projects, I just didn't have any other possibility, and I just tried to learn it. And then I would also thank my supervisors, especially Professor Brück, who definitely played a role in encouraging and showing the example as well, how to code and as part of a clinical work. And yeah, I would say that having somebody as an example, or somebody close to you that is a colleague, or it helps a lot. And really to encourage further, I think there are huge resources in the internet. It's really easy. I think the only thing you need to know by now is just how to Google, and after that, I would say that everything else is then you can find the information there.
That's fantastic. Thank you very much, Oscar, for taking your time, and once again, congratulations on your publication, and wish you very much luck with your future endeavors in implementing more advanced machine learning methods for hopefully for implementation in the clinic also at some point. It's been a pleasure talking to you today.
It was a pleasure. Thanks, Lars.
Thank you. See you.
See you.